My name's Julie Cohen. I'm a genetic counselor. I'm at Kennedy Krieger in Baltimore. Among a variety of roles, I am the genetic counselor for the Moser Center for Leukodystrophies. I've had the pleasure of being at Kennedy Krieger for almost 15 years and with the Leukodystrophy Center for about 10 years. I'm happy to give a talk like this every year. I don't want this to be a lecture. I have some slides and I can go over some background information, but I'd really like to tailor this as much as possible to the people here in the room. And I'd like to facilitate a conversation, as you can tell by being a counselor. I want to counsel and I want to talk with all of you and make this as useful and personalized as possible. So I'd love if we could start by going around the room. If you could tell me your name, your interest in being here, if you're a family member or someone with a leukodystrophy, if you feel comfortable sharing your diagnosis, if you're not a family member, if you're here from academia or industry, just telling me that background as well. So I'm going to start here with these lovely ladies.
I'm Heidi Edwards. I'm the president and founder of Sisters Hope Foundation.
Okay.
We support those affected by ALSP.
Great.
And we've met, Julie, I'm Cynthia Cassandro. I'm with Digital Neuroscience.
Wonderful.
We have a program in development for ALSP.
Great. Okay.
My name is Elizabeth Calderon. My son Dante is 5. He has HABC leukodystrophy.
Okay. 5 years old. Great age. Yeah.
I'm Paula, and I'm a researcher at the Von Haaren.
I'm Mandy. I'm with BridgeBio. As with Therapeutics, we have investigational gene therapy in cannabis disease. Excited to learn from all of you.
Julie, you might pass the mic around because
Yeah.
we're not getting the...
Oh, sure. Okay. I'll be Vanna White here.
Okay. Mandy with Aspa Therapeutics. I work on patient advocacy, and we have an investigational gene therapy in Canavan disease, and I'm glad to be here to learn from you and all of you. Thank you.
Okay. Pass it this way.
I'm Josh. My daughter is 13 years old. She just got diagnosed about 4 months ago. It's a very rare one, so we're not quite sure. We're just here trying to figure out what's going on. So,
Hi, I'm Abby Cook, and I have 2 sons with cerebrotendinous xanthomatosis, CTX, and I'm very, very interested to learn about the genetics involved in this and other leukodystrophies.
Hi, I'm Michelle Willette. I work for Ionis Pharmaceuticals, and we have a neurology pipeline with one product under investigation for Alexander disease.
My name is Pedro. I'm on the board of Cure CADASIL. I myself have been diagnosed with CADASIL and I have a 4-year-old, and so here to learn.
Hi, everyone. My name is Shay. I am a genetic counselor, actually, but I work for a pharmaceutical company that has a treatment for cerebrotendinous xanthomatosis.
Love that. Love this connection. All right.
Hello, I'm Nicholas Carvelas, a postdoc at the Elahi Lab. We're working on CADASIL, so we're very interested in learning more about the patients as much as genetic counseling as well.
Yep. My name is Ryan. I'm also a researcher at the Elahi Lab at Mount Sinai Hospital. We're all working on CADASIL.
Hi, I'm Lisa. Same, working in the Elahi Lab in Mount Sinai. I'm a researcher there.
I'm Ruth. I'm the same as the rest of the row.
Yeah, me too. I'm Pauline. I work at the Elahi Lab too. I'm so glad to be here today.
I'm Abby. I'm a carrier of adrenomyeloneuropathy, and this is my dad. So.
I'm Brad Bauman, and it's my daughter. But I have AMN, and which probably was diagnosed 20 years ago. We'll probably hear more for her today than anything. So.
Okay. All right. And last but not least.
I'm Jane. I have a son-in-law who has CADASIL, and he and my daughter have 3 children, and because those 3 children have a 50% chance of having inherited it, I'm very interested in the genetics and advising when you do that with children.
Yeah, that's a big question, is, testing children. The questions I imagine are in the room. What caused this? Why did this happen? Right? So what caused my leukodystrophy, my child's leukodystrophy? Why did this happen? There's nobody else in my family. What do you mean it's genetic? could anybody else in my family have it? Could my sisters, my brothers, my grandparents, does anybody else need to be tested? Should I have genetic testing? My diagnosis is 20 years ago, just on a biochemical test. Do I need a genetic test? What do my genetic test results even mean? So these are some of the many questions that I face, or that my patients and my families face when they come to meet with me. And there's lots we can talk about today. Like I said, I don't want this to be a lecture, but I think because we're all coming with different backgrounds, I'm going to kind of go over a couple slides just reviewing some of the basics about genetics and about inheritance patterns and chances for relatives. And I'm going to talk also about family planning or reproductive options. And I want you guys to interject and ask questions, and we can direct the conversation. Oh, and I forgot to ask about our people on Zoom. Greg, my friend, anybody on Zoom wants to chime in with their backgrounds or diagnoses? Okay, all right, okay. It almost makes it a little bit easier for me to just engage people in front of me. What questions, in addition to kind of questions I put up on the slide, anybody want to shout out a question that they want to make sure that I address today that I can weave through? Yeah.
So we just got back the genetic testing for my husband and myself.
Yeah.
I apparently supplied the likely pathogenic allele, and he supplied the unknown.
Okay.
Unknown significance. So we have two sons.
Yeah.
That have this. So is it now going to flip that of unknown significance into the likely pathogenic?
All right, so that's a question kind of related to the last bullet. What do my results mean? What is a variant of uncertain significance? What is pathogenic? What's a mutation? So, okay, that's good. I'll keep that in mind when I get a little bit later in the talk. Okay, so everything you need to know about genetics in less than 30 seconds and in one slide is the cookbook analogy, okay? So our genome is a cookbook to make a person. Genes are those recipes in our cookbook. A gene is a section of genetic information that's the instructions to make something. We have 20,000 genes, 20,000 recipes in our cookbook. DNA, that's the letters or the alphabet that spell out all of our recipes. Chromosomes are like chapters that organize all of the recipes. And proteins, so we talk about genes making proteins, that's the finished product, that's the cake, right? Everything you need to know about genetics, and we're going to use this as we go further through. So our cookbook, we actually have 2 copies of our cookbook. We get one from mom and one from dad. Most of our chromosomes, most of our chapters are the same by sex, males and females. But the last pair of our chromosomes, so we got 46 chromosomes total, 23 pairs. The last pair are the sex chromosomes. Ladies have XX, men have XY. All right. So I'm going to talk about inheritance patterns. So most leukodystrophies are caused by changes in single genes, so a typo in a specific recipe. They're as opposed to changes at the chromosome level, although there are some variations there. So most are caused by variants by a specific typo. So like I said, we inherit 1 copy of each of our chromosome pair from mom. So 1 copy of our genes from mom, 1 copy from dad. And there's different inheritance patterns. And it's like flipping the coin, what you're going to get from which of your parents. 50-50 chance of which copy of the gene you're going to get. So I'm going to start by talking about autosomal dominance, autosomal dominant leukodystrophies. So CADASIL, that's an example of a dominant inherited leukodystrophy. So dominant, that means that the condition manifests or shows up when 1 copy of the gene is altered and not working. And it's the same. It doesn't matter whether you're male or female. And so for a person with CADASIL, there's a 50% chance in each pregnancy that the child or the offspring would get the abnormal gene and develop the condition one day, and a 50% chance they'd get the normal gene copy. And like I said, it's flipping the coin, right? So in this little diagram, we see 2 out of 4 kids having the condition and 2 not, but it could be all 4, it could be 1, it could be none. So it really is just that flip of the coin. So questions about dominant inheritance. So it can be kind of tricky for some conditions where it may not show up until later in life, or there may be variation in the symptoms. some dominant condition. So a dominant condition can be inherited from a parent, or sometimes it's a new mutation. So it's a genetic variant that occurs new in that affected individual. So neither parent has the abnormal gene or the typo in the gene, and just by chance, usually in the egg or the sperm, a mutation or a change occurs, and then the child has it. So the recurrence risk for these de novo conditions, so for parents who have a child with a de novo variant, it's a low chance to have another child with it, but it's not zero because of something called germline mosaicism, which means that the gene variant could be present in just a subset of the parent's egg cells or sperm cells, but not anywhere else in the body. We don't have good numbers on that, so we usually say it's around like maybe 1 or 2% chance to have for recurrence of another child with a de novo mutation. And let's see what else. So when we have the child has a de novo genetic variant, other people in the family like aunts or uncles or cousins, they're not at risk to have it. It's really just that child. But the child themselves then could pass it on. So the next we'll talk about is a autosomal recessive condition. So examples here, we have CTX is a recessive condition. So this, in recessive disorders, the condition only shows up if both copies of the gene are altered, and an individual with one altered gene copy is fine. They don't have that disorder because the backup copy is normal. So typically in a recessive condition, when a child has a recessive condition, both parents are carriers, and in each pregnancy they conceive together, there's a 25% chance that the child would inherit the abnormal gene from both parents, and that's shown in the little purple on this side of the screen. And then there's a 50% chance in each pregnancy that the child would just be a carrier but not show symptoms, and a 25% chance that they'd be not even a carrier and unaffected.
Julie?
Yes. Yes.
This is from Emily. The question is this: if the parent has CADASIL and the child does not, are the grandchildren in the clear?
Oh, great question. Yeah, yes, I'm going to restate the question. Yes, thank you for that question. So the question is, if a parent has CADASIL, or let's say another dominant condition, right? Let me go back to that slide. So if a parent has CADASIL or ALSP or another dominant condition, but their child doesn't, and the child's been tested, and so we know for sure they don't have it, is that child's children, so the future grandchildren, at risk? And the answer is no, because gene variants do not skip generations. So if that, for a dominant condition, if that individual, so I'm gonna so this individual, unaffected, they can't have a child with it unless it were a de novo mutation. I mean, I'm not going to get into those rare things, but basically they can't pass it on. So great question. For some conditions, it could appear to skip generations if this is a condition where maybe it's late onset and the symptoms don't show up until much later, or that person, passes away before they develop the symptoms. But but testing negative, you can't pass it on. Other questions on that topic? All right. So I remembered something I wanted to say about de novo variants. So I'm giving the example here for a dominant condition that it can be a de novo variant or mutation. That can also happen in any inheritance pattern. So for X-linked conditions, which I'll talk about in a second, like AMN or adrenomyeloneuropathy, sometimes those mutations happen new in that individual as well. So really any genetic variant it has to start somewhere. It starts somewhere. Okay. So we were talking about recessive, right? Okay. So other people in the family, like let's say the parents who are carriers, their siblings, they could be carriers, because typically with recessive conditions, it goes back generations. We just have never seen it before. It doesn't show up in the family until two people who happen to be carriers of the same thing meet each other and have a baby. That's the only way we know about it. And every single person in this room, all of us, everyone on the planet, we're all carriers for multiple rare recessive genetic conditions. We just don't know it. All right. So now I'm going to talk about X-linked conditions. So these, oh yes.
Dominant and recessive.
Yes.
So with ALSP, a mutation on the CSF1R gene.
Oh, back it up. Yeah.
Okay. So with ALSP, there's a mutation on the CSF1R gene, which causes ALSP. But there's a mutation on the CSF1R gene which causes a recessive form also called Bandos. So we have dominant ALSP, recessive Bandos. When you for example, I am not a carrier, but my two sisters were and they passed away from ALSP. We have the dominant form in my family. How do they know in the lab that it's not the recessive form? It's not really Bandos.
That's a great question and perfectly exemplifies why genetics is not as simple as these pretty little drawings, right? So there are some genes where variants or mutations, I'm going to use those words a little bit interchangeably. I hate the word mutation because it sounds like swamp monster. And actually in the field of genetics, we're supposed to get rid of that from our vocabulary, and we use terms like pathogenic variant. This is a little, I'm kind of going on a tangent to explain my vocabulary. So pathogenic variant or likely pathogenic variant, that means a DNA change that's going to cause a disorder, what we used to call mutation. So I may use those interchangeably just to try to be clear because pathogenic variant is a mouthful. Okay, so back to your question. So there are some genes where variants or mutations can be, or those genes are associated with dominant or recessive inheritance patterns. And how can that be? So it could be differences in the effect of the variant on the gene. So here we go. So the typo in the recipe, is it causing the recipe to just be absolute junk and not make any sense? That may be one type of variant. Or is it the typo is causing too much of that protein, of that product to be made, right? So a typo in a recipe, a genetic variant can have different effects on the protein that's made and then therefore downstream. So that's one reason why different variants in a gene could cause dominant or recessive. Does that kind of make sense? But then your question is, well, how does the lab know? Yeah, it's really hard. And it's totally dependent on the gene. Sometimes if a disease is well known enough, where we've seen these sorts of variants enough, we have enough cases, and we can say, oh, if it's this type of variant, it's going to be recessive. If it's this type of variant, it's going to be dominant. The leukodystrophies, we're having newer forms discovered all the time. We're also learning new things about conditions we thought we knew everything about. So when the CSF1R gene was discovered as the cause of ALSP, we thought, oh, it's only a dominant thing. And then more recently, there are these other cases. So genetics, we like to think we know everything. We really don't. There's a lot we don't know. So it can be challenging in the lab's case. I think for something like ALSP, it's probably a lot dependent on the clinical presentation of the individual. So that's one piece that comes in, and it may be also variant specific. I don't know enough about the genotypes for ALSP to answer specifically. Yeah. Thank you for all these questions. Okay, so now we're going to talk about conditions on the X chromosome. So in an X-linked condition, males or people born with an X and a Y chromosome, they will always be affected or manifest that disorder because they've only got one copy of the gene. They don't have a backup. Females or individuals with two X chromosomes, they've got two copies of the gene, and they may or may not be affected. We used to just call females carriers, and we still use that term, a female carrier. That sort of can imply that a female will never develop the condition, but we know for many conditions like adrenoleukodystrophy or adrenomyeloneuropathy, Pelizaeus-Merzbacher disease, which is also X-linked, that women can show symptoms. It may be milder, it may be later onset, it may look a little bit different than the men. And this is an important counseling point, I think, because women may not realize that. They may have symptoms and are dismissed by doctors as, well, I don't know what's going on with you. You just, you're taking care of a kid with disabilities. Of course your feet hurt, right? So we're really trying to increase or empower women and increase knowledge that women who are carriers or who have an X-linked condition that they may show symptoms, that way they can take control of their own health and advocate for themselves. So another word we use here, especially in the ALD community, is heterozygotes. So a woman is an ALD heterozygote, which is a technical term to mean that one copy of the gene is altered, the other is normal, trying to sort of take away that carrier implication. Okay, so that's an X-linked condition, and now we'll talk about chances of passing it on, which differs depending on the sex of the individual. So for a male with an X-linked condition like ALD, like your dad, his only copy of the gene, his only copy of the X chromosome has that typo. So all of his daughters are going to be carriers because if it's a daughter, she got his X chromosome. So all daughters would be carriers. No sons would be affected because the sons would have inherited the Y chromosome and a presumably normal X chromosome gene from the mom. So for a woman who is a carrier or heterozygote, this is showing on this side of the screen, ... So in each pregnancy, again, it's a flip of the coin, whether the child's going to inherit the normal gene copy or the abnormal one. And then depending on which chromosome they get from dad will be the outcome. So in each pregnancy, it's a 25% chance to have a son who's affected, 25% chance to have a daughter who's a carrier, and then a 50% chance to have a child of either sex who's unaffected and non-carrier. So a misperception or a confusion in X-linked conditions is thinking that it only comes from the mother's side. So a woman could, who is a carrier or who has an X-linked condition, could have inherited from her dad or her mom. And again, we can see these go through many generations of the family. Questions about any of these inheritance patterns or risks or chances for family members? Okay, all right. So the next section of slides I have is talking about reproductive options or family planning options for an individual who themselves has a leukodystrophy or parents or relatives of an individual with a leukodystrophy. And when it comes to having children and having a family, there are a whole many ways to do that. And these are options that individuals consider. One of which is going about it the old-fashioned way, natural conception, and maybe choosing to have the child evaluated after birth if it's an early onset condition, or letting the child make that decision later in life for a later onset condition if and when they want to know for themselves. There's the option to do natural conception and test during the pregnancy, and I'll talk a bit more about that. There's the highest tech option to do in vitro fertilization and test embryos for the condition prior to starting a pregnancy. That's called PGT. And then there's alternatives like adopting, using donor eggs, sperm, or receiving a donated embryo, or having no additional children. So these are the range of options, and I'm going to talk about a couple of them in more detail. So pre-implantation genetic testing, or PGT. So this is genetic testing of embryos that are created through IVF to determine if they have that genetic condition. It's incredible. So this is a picture from under a microscope. So this is an embryo, and this is, that's a pipette, and this is a little, or maybe the other way around, a little doodad. So what this is showing is, so this is about 5 days after the egg and the sperm have been fertilized. And that is about, maybe a couple hundred cells big, and a few cells are plucked off of each of those early embryos, which doesn't damage the embryo, and those cells are tested. It's pretty wild that this can be done. So PGT for a leukodystrophy or a single gene condition, that's called PGT-M. And the advantage of PGT is that it allows selection of an unaffected embryo, what would be an unaffected child, to use for pregnancy, so to start for pregnancy. So I'll talk a little bit more about what that process looks like. So in vitro fertilization, so what this process looks like is before this is even started, before you even go into the fertility clinic, there's lots of tests that are done to check a woman's fertility and the man's fertility. Genetic testing, because we need to know the specific genetic mutation. We need to know the type of leukodystrophy, and we need to know the exact genetic variant. We need to know the status of the parents and sometimes other family members. And a custom, it's called a probe, is created for that particular couple. So a specific probe that's going to be used for the cells from the embryo to check for that mutation. Then the process of IVF, where the woman receives medications, injections, and oral to stimulate her body to produce many eggs. And then those eggs are retrieved through a minor procedure. The male partner provides a sperm sample. In the lab, the little spermies and the little eggies, put on some nice music, and they kind of get their thing on. And so then we have some fertilized eggs, and then those eggs those fertilized ovum are allowed to grow in the lab for about 5 to 6 days. And that, I put the star here. And then so that's typical IVF. And then at this point in the process, the embryos are biopsied. And so those few little cells are biopsies and sent off to a highly specialized genetics lab. And then those embryos are frozen, flash frozen, which incredibly doesn't damage them. And they're frozen because it takes a couple weeks for those genetic results to come back. And then the lab kind of sends a report, says, okay, embryos A, B, D, G, whatever, are normal or unaffected. They don't have the mutation. And then in a subsequent cycle, the embryos that are selected are thawed, and 1 or 2 of them, depending on the situation, would be transferred to the woman's uterus to start a pregnancy. So PGT it has a lot of pros, right? The number one being is that this is sort of before pregnancy, right? So the baby isn't already in the womb, and we're not talking about doing prenatal testing and potentially terminating a pregnancy. PGT is very accurate. It does still require confirmation during the pregnancy. Some of the downsides of PGT or limitations is that it's expensive and it's not always covered by insurance. It is, a process to go through for the couple to go through all of this IVF process. PGT, another limitation of PGT is that you have to know that specific diagnosis. So for leukodystrophies, we have a lot of families where they don't know. They do all the fancy testing, right, and still don't know the specific diagnosis, and it can't. So we can't do PGT there. PGT can't be done for if there's a child with a de novo mutation and the parents don't have the mutation. You can't do PGT for some complicated reasons that I won't explain unless anybody really wants to know. But really, it has to be something that's inherited, and you need samples from family members. So those are some of the pros and the cons, but it is an option that a lot of couples would like to do if possible. Yeah. Questions about PGT before I go on? Okay. So another option is prenatal testing. So this is genetic testing during a pregnancy. So natural conception, and then there are 2 procedures that can be done during the pregnancy to specifically test the fetus, and that would allow the option of pregnancy termination if the baby is affected, or to use that information to prepare for the birth of that child, especially for an early onset condition where that information could really be important for the families to know what to expect. So the 2 prenatal testing procedures, one of them is called CVS, not the pharmacy, chorionic villus sampling. So this is performed at the end of the first trimester, so about 11 to 13 weeks after pregnancy has begun. And it's done under ultrasound guidance. There's a little catheter that's put in through the cervix to take out some of basically the placenta tissue. And that is sent to the genetic testing lab to check specifically for the gene mutation. Another procedure, it's one or the other. The other procedure is an amniocentesis or amnio that's done a little bit later in pregnancy, after 16 weeks. It's the same idea, which is getting some cells. In this case, it's amniotic fluid with a needle through the abdomen, and those cells, again, are tested for the specific variant, or sometimes a biochemical test can be done. So these are highly accurate tests, so that's a pro. But some limitations are that, again, you need to know that specific diagnosis, and usually you'd also need to know the specific mutation. And then another just downside why this is a tough decision for a lot of couples is that you already have an ongoing pregnancy. So those are prenatal testing. I get questions a lot about whether can we do this on like a blood test of the mother, because now there are some screening tests that are offered to pregnant women where they can screen for like certain chromosome disorders in the blood. That is not yet possible for single gene conditions. It could be in the future. There's a lot of industry and science working on this so-called non-invasive prenatal testing by looking at the blood of the mother, but it's not there yet. Yeah. Oh, actually.
Oh boy. Is there one as an expert that you would recommend over one another? Is it specific to like the type of mutation that you're looking at? Because there's of course going to be risks associated with this. Yeah. Is there one that's preferred over the other? Of the two. Yeah.
Good question. So the question is there one of these procedures that's preferred over another? So thank you. One thing I forgot to say of another, that reminded me, another downside of these procedures is that there is a small risk of miscarriage. It's about maybe 1 in 200 to 1 in 400. So a small chance, but not a zero chance. So which one is preferred? I think generally the CVS is preferred because it's earlier in pregnancy, and so you're going to get results earlier. But results can take, depending on the lab or the test, it could be 2, 3, 4 weeks. Yes, another question.
Is that even working?
I don't know. I think it is.
Is it? Okay. I was just wondering, are there any requirements, or is anyone allowed to get prenatal testing?
So question is, are there any requirements? So prenatal testing there's no requirements. Anybody can have it. There may be limitations potentially on insurance. Again, lovely American healthcare system. There could be access issues though. Right? So these are specialized procedures, so not just any obstetrician can do them, but in theory they should be available to all. Yeah. No, don't apologize. I love the questions because I don't just want to be up here and talk at you.
So amnios are fairly common. You hear a lot of like couples like talk about, should we have an amnio or not? And so it screens for, from what I understand, like a bunch of different things. So when we're talking about leukodystrophy, something more rare, you're saying that we have to know like the actual mutation from, and it can't be de novo again. So it would have to be something that's inherited. So is that because it's not typical and it's not on the panel? Okay, there it is. Sounds good. Yeah.
So you're filling in the questions of the things that I didn't put on here. So I get another question I get is, well, why can't you just test for everything? Either maybe in a carrier screen, right, even before someone gets pregnant, or in one of these procedures. So you have to know what you're looking for. So these procedures are commonly offered for not related to known genetic conditions in the family, and you can test for chromosome disorders, you can test for Down syndrome, you could test for any genetic condition in a prenatal sample. You can do whole genome sequencing or look at the entire cookbook, but you have to know what you're looking for. So for a leukodystrophy, yes, you'd have to know what you're looking for. Now they can, with prenatal testing, because it can be done for couples who have a child with a de novo mutation, they can still check for that mutation in that sample because they can directly test for that mutation. Yeah. Another question I get from couples who have a child with a leukodystrophy is especially if it's something that is inherited, right? That turns out that both parents are carriers of. Well, why didn't I know this? Like, why, I had genetic testing before I got pregnant, or why couldn't have somebody told me that I had this? So it's not routine to screen or test the whole population, anybody in the population, to know every single thing they're a carrier of. Again, it's technically possible, but it's just not offered. There are broad carrier testing panels that are offered now to all couples that do include a number of leukodystrophies, that include ALD, that include a number of rare leukodystrophies, but there's limitations to those as well. Yeah, so often this comes as a surprise to families when this is diagnosed. Yeah.
I just have a question. I know there are.
I'm getting my steps in.
23andMe and some of these different types of options available that are quite accessible for people in the community. Can you just talk a little bit to the pros and cons of those and the usability of those for couples who are considering?
Yeah, so direct-to-consumer genetic testing, because now you can, through 23andMe, through Ancestry, through lots of different labs, you can get your whole genome sequencing, your whole genome sequence. So that is genetic entertainment. So if you want to do that, I mean, I'm not trying to, it is sort of, whatever. It is. So that's cool and that's fine, but that is not a way to get health-related information because those labs are not giving, for the most part, health-related, like a validated report. They're not checking for everything. They may issue some reports to know, oh, you're a carrier of this, that, or the other, or you have the Alzheimer's, ApoE, variant. But that's pretty limited. Now, there are then third-party companies where you can take your sequence data and upload it, and they will spit out all of your cajillion variants, and then you could, try to match that up. But that's not clinically validated, and a lot of those variants and people come to us in the genetics clinic and say, oh, my 23andMe, I looked at the data. It turns out I had somebody, actually, it was a leukodystrophy, actually, that they came to us thinking, my child has this leukodystrophy because I saw it in their raw data. Well, when you send that, a lot of times if you send that to a CLIA certified or a clinical lab, it's not real. It's a false call. So my advice there is if you want to do that for fun, and if you're comfortable, and if you've read the fine print and are aware that for some of these companies, your data might be sold, that's cool. But if you're needing a diagnostic test, if you are needing carrier testing, you come to see a professional, and we can do it in a proper lab. Yeah. So quickly to mention a couple other options that some folks don't really think of is using donor eggs or sperm. So why would you do this? So an individual who has an autosomal dominant or an X-linked condition and maybe the PGT is not possible or not accessible, maybe prenatal diagnosis is not acceptable to them, so they could use a sperm donor or an egg donor and still have them or their partner carry the pregnancy. Couples who are both carriers of an autosomal recessive leukodystrophy, again, maybe those other options aren't accessible or acceptable, and so using a donor who isn't a carrier, and there may be other scenarios as well. Also for, and then receiving a donated embryo. So this is something as well. So maybe folks are comfortable with IVF, with that idea of IVF, but aren't comfortable with creating embryos that would not be used and potentially be destroyed, right? So taking a donated embryo and using that to try to have a pregnancy. So those are options.
Julie, we have another, this is kind of a question and comment.
Yeah, oh, actually, yeah, no, you say that, I'll repeat it, yeah. Yeah. So thank you for sharing your experience. So that individual and his partner had a child with MLD, metachromatic leukodystrophy, which is recessive. And then in a subsequent pregnancy, they did CVS testing, and the baby was predicted to be unaffected. And then after birth, confirmed, which is also always a good idea, to confirm postnatally, the baby was unaffected. And I'm so glad to hear that. Yeah. Yeah. And so couples and families choose all of these options or make different choices, right? And there's no right or wrong. There should be no judgment because there's so much that goes into this, even like the numbers, right? A high recurrence risk, a number that's a high chance, 25% chance to one person, that same number might be not high to another person and vice versa. So the last slide I have on this section is about adoption. And I want to give you, this is, I think, a great website for Resolve, which is a fertility organization that talks so much about all the different types of adoption and how to navigate that process. So that is the end of the main part of my slides. I have some resources, my contact information. I have a couple things we can talk about genetic tests, but I just want to make sure we have plenty of time, and we do have time for questions, other questions, especially from any of our patients or families in the audience. Questions that have come up for you. Yeah. Oh, I guess I just educated you all so well. Yes.
When you talked about men getting one gene and women getting two, that's often with women it's more recessive, if I understood that correctly, that the disease could be less prominent. Yeah. So in my family's case, my son-in-law has CADASIL. His mother would have been the carrier. She died in her 60s, but not from that. She died from a pulmonary embolism. But her father had been also the carrier. So would that mean that she didn't show signs of CADASIL because of having that more recessive kind of trait?
Yeah, I'm glad you asked that, and that illustrates that this is kind of confusing stuff. So for CADASIL, so that is a dominant condition, and dominant conditions, genetically speaking, the chances don't differ by sex. So for a dominant condition, women, males and females have an equal chance of inheriting the mutation and being affected. And so there shouldn't be a sex difference with expression. So we may kind of see patterns in families where it seems like, well, most of the women really had milder symptoms, or maybe they didn't show it, but it wouldn't be, I think that's just kind of a coincidence in the family. So what you were saying, because I think what you were thinking also about an X-linked condition where it actually does differ by sex. So what you were explaining, that the mother really died at 60 of some not of CADASIL, and she never really showed symptoms. So why is that, right? It could be that her disease, had she lived longer, it would have shown up, because CADASIL is something where there's a big spectrum in terms of age. It could be that she had like super mild symptoms that wasn't recognized to be CADASIL. So a lot of these leukodystrophies have this wide variability, and that can also make it challenging for couples and families and individuals, because usually based on the genetic result, we can't predict, oh, this is exactly what this person is going to look like in terms of their symptoms. This is when it's going to start. This is how fast it's going to progress. There's other factors that play a role. Yeah, sure. And going back to the X-linked condition, so those are where women, because they have a backup copy of the gene, they may not show symptoms. Why is that variability? But some women do show symptoms. It can be complicated genetic reasons. It could be, again, other factors that we just don't necessarily know about yet. Yeah, yes.
Thanks. I had a question on the challenges in family dynamics, maybe between sibling testing.
Yes. So testing in families, right? So these are, for the most part, family disorders. And people have different feelings and opinions about whether or not to get tested. I think especially for conditions that start later in life, like CADASIL, like ALSP, and conditions where there may not be any treatment available right now. So do you want to know, right? Do you want to know if you are likely to develop a significant neurologic condition when you're older? Maybe you do, maybe you don't. And it can be different in family members, right? You can have siblings feeling differently. And again, there isn't a right or wrong answer there. And individuals, there's just so many factors that they consider, like what would that information mean to them? What's the value of knowing they don't have it? What's the value of knowing they do have it? What kind of coping mechanisms do they use? And sometimes when you get multiple people in the room, you can have difference of opinions, but again, everybody's unique. And I really encourage folks not to rush into making that choice, whether or not to get tested. Someone also asked the question of testing children. So testing children for conditions that aren't going to start until adulthood. This is a point of lots of ethical discussion in the field of genetics. Again, we have to weigh different factors and there isn't necessarily a right or wrong answer. It used to be, the old rule was, well, you never test a child for an adult onset condition unless you're going to change something about their treatment in childhood. Like 18 is the magical number. Well, nothing magical happens at 18, does it? I will counsel teens who are at risk of being a carrier for an X-linked condition, and they may be mature enough to really articulate, I want to know this now. It may not be relevant until I'm ready to have kids, but now is the time to know it. I've thought through what I'm going to feel one way or the other. So we don't take that magical 18 number rule. There's considerations with could knowing a child's status affect things like insurability down the road or jobs. There are protections in place for health insurance, for jobs. There are limitations in those protections for other types of insurance. We also take into consideration family dynamics, right? If the parents are losing sleep over their child's status and it's causing such distress for them, and that distress can't be resolved through other sort of counseling or coping mechanisms, maybe it is appropriate to test the child. So again, it's not just black and white there. Mm-hmm. Yeah.
Just one question that kind of came in my mind a little bit, and I don't know how big of a question it is, but I've been diagnosed AMN, X-linked, and there's really no reason to have my daughters tested, correct? I mean, you're talking about testing, so I kind of was wondering, but there's really no reason to even test at this point with them because they are, correct?
Yeah, that's a good question. So do someone who we know must be a carrier or affected, do we need to test them? In a situation like AMN, that's X-linked, so we know that daughters are carriers. You don't need to test, I think, unless it's going to be using any of these reproductive options like IVF and PGT or prenatal testing, because we are just going to want to confirm the presence of that genetic variant. To a question for conditions that are diagnosed just based on, say, an enzyme test, or a biochemical test, it is still important when possible to do the gene test to identify that specific variant, to allow accurate testing of family members, and to allow those family planning options. Yeah. Mm-hmm. Yeah. Let's see. I get questions about types of genetic testing. This question of, are all tests the same? Or I already had a genetic test. It was negative. We didn't find the answer. So there's a lot of different types of genetic tests, and they're different, maybe appropriate in different scenarios. So when there's a condition already known in the family and we know the exact mutation or variant, then we may, if we need to know someone else's status, can zoom in on that specific variant, that known familial variant, and test for that instead of looking at the whole gene. In the diagnostic process, you know, some of you here had children who were diagnosed. I imagine there was some odyssey leading to that or some process. And if there's a specific condition that's suspected in the individual or themselves, then it may be a focus test. So, you know, the MRI looks like CADASIL or the MRI looks like ALSP, so we'll zoom in on that specific gene and check that. Sometimes it's a test that's looking at many genes, like related to leukodystrophy, or we have tests that can look at all of the genes or the entire genome. So there's different levels of testing, and the results can be complicated when they come back. We had a question about variants of uncertain significance or a likely pathogenic variant. So kind of interpreting, is a particular DNA misspelling, if we've never seen it before, is that going to cause the disorder or not? And there's a lot that goes in on the lab side. If you have genetic test results that are not straightforward, genetic counselors can help with that. Yeah. Mm-hmm. What other questions for Ask the Genetic Counselor? We're almost out of time, but I am available. Mm-hmm. All right. Well, I'll hang around here if you guys have specific questions that you want to come and just talk to me about. But thanks for coming and for listening in and for participating. Good. And I'll leave my contact information here. My email, you can reach out just to me. That's the email for our Leukodystrophy Center if you're interested in coming to see us. And if you need to find a genetic counselor, we have a handy website called findageneticcounselor.com where you can locate a genetic counselor near you.